Welcome, friends, to our next topic, which is muscle. Skeletal muscle, smooth muscle, cardiac muscle. It's a very interesting topic, so just follow me and we'll go discovering what we need to know. Let's start with skeletal muscle. And we'll start in different levels. We'll start with a simple muscle, like for example, the biceps has one tendon, and in the upper portion it has two tendons. But for the most part, it's like fusiform. And once you look at it, it is composed of numerous cells, skeletal muscle cells, also called fibers, which is a name I don't like too much, because it may be confused with other fibers in the same muscle. The muscle cells go all the way from one extreme to the other. So in other words, they are very, very long cells. For example, even in the most muscle of the thigh, the sartorius, for example, muscle cells may reach about more than 30 centimeters. It's huge. And on top of that, the cells are multinucleated. We just look at, by just looking at the cell, the muscle, you will find out that what you see on the surface is mostly connective tissue. In other words, the muscle is not alone. It is tightly packed. It's well protected by connective tissue. And also we have a connective tissue at the ends, the areas of attachment to the bone. In this case will be the shoulder, those ones. Now, if you want to know more about the muscle, you will have to get perhaps something like a knife and cut the muscle here in the center or somewhere like that. If you do that, you will have something like this. Something like this, just like when you cut meat from the supermarket. Now you're going to see the cells here, but in order to see them better, you would rather look at them this way. So I would rather recommend you to rotate the muscle so that you have the tendons here and the cells. Now, when we study muscle, we're going to have to simplify, because muscle is a little bit complicated and there are numerous structures involved. So we're going to simplify a little bit. So when you cut the muscle like that, you see the cells that have been cut in a transverse fashion, and they are going to pretty much, they're going to look like polygonal or ovoid, and they're going to be red, and they're going to be eosinophilic when you cut them in any direction, because muscle has numerous filaments of protein, actin, and myosin, which are proteins, and they are eosinophilic. So now the other thing that you see immediately when you cut a node is that the numerous muscles, the numerous nuclei, are at the periphery. Sorry about that. They are the nuclei are very eccentric, almost at the edge of each individual cell or cell muscle. Now, we said that the muscle is covered by a layer of connective tissue, And we can actually make it evident, clear, that this is the layer of connective tissue. It's like a sheet of connective tissue that is going to be called epimysium. Epimysium. And it doesn't end like that. It will have also different groups of fibers being surrounded by a layer of connective tissue, which is called the perimysium. The perimysium, I'll just put it for you. Perimysium, and around each individual fiber, there is also connective tissue that is going to be called the endomysium. Those are Greek prefixes that will help you. Epi, it's around. Peri, it's around each fascicle. And endo is within and around each individual cell. Now, you will think about what else do we have in a muscle. Of course, we have the possibility of having, or we have to have, arteries and vessels that mostly go in a longitudinal fashion. And the capillaries will also go in a longitudinal fashion. Now, when we want to go and study each individual cell, we'll have to simplify, and we will take only one cell, just one single cell. Bear with me. And one cell, what is the shape of a muscle cell? The shape of a muscle cell is cylindrical, and it's very long. So, in other words, a single muscle cell will go, if we can go this way, it is cylindrical, roughly cylindrical, and it is completed at the ends by the connective tissue from the tendons of attachment. Now, if you start looking at it, you start realizing that, let me see here, we have to start nuclei. The nuclei are going to be multiple and located at the periphery. So it is a multinucleated cell. It is a cell with multiple nuclei. It's not the only one in the body. Another one that comes to mind right away is the syncytial trophoblast from the placenta, which has a humongous large number of nuclei. Anyway, this is a multinucleated cell. How did we get to have a multinucleated cell? Because the cells that start in the embryo are called myoblasts, attach end to end to each other and form these tubules that are going to later be skeletal muscle. Okay, now is there anything else important here? Yes. When you look at it on a microscope, the cell, it's not totally homogeneously red, but it has striations. In other words, it is a striated muscle, which is the most important striated muscle, if you want. In other words, the muscle has these cross striations, showing one of them dark and the other one light. This is an oversimplification, but definitely showing to you that the skeletal muscle has these striations. Why? We're going to try to find out. And in order to find out, we will have to kind of look at a portion of the cell. Okay, in other words, we already looked at one cell, and now we're going to look at only one portion of this cell. And if you, let me look at it, if you use only, let me see, use the red. If you use I'm going to use then black. If you're going to enlarge this area only, we're going to have a cell that goes this way. We're cutting the cell. And that has multiple nuclei, as we know that. And the nuclei are going to be, let me use another color. We'll have the nuclei placed at the periphery. And obviously, we're going to have numerous striations that are going to go one way, where I go totally, I'll try to make them as broad as I can. These striations that are. Now, what is the cause? What causes these striations? Well, these striations are the result of striations in multiple units called myofibrils. And I'll use the black marker for that. The myofibrils will come up. I'm going to oversimplify like this. Maybe like this. I don't want to draw too many because it's not easy to, there are just too many. And you could have more cut in this direction. Now, what happens to these myofilaments? I'll write it down for you, myofibrils. These myofibrils have cross striations. And so if I go back to each one of them, it will have this appearance. Each one parallel to each other, and it's going to be really interesting. So if you hold, if you want in your imagination, a skeletal muscle, it's like holding a pack of cigarettes and showing it to somebody. The myofibrils will be the cigarettes, and all of them are stained in the same fashion vertically, if you try to understand it. So these are the myofibrils. Now, what are the myofibrils composed of? You have to go the same way like that. They are composed of myofilaments of actin and myosin. So in order to understand what goes on with a single cell, let me correct a little bit this nucleus, which is a little bit kind of crooked. We're going to try to understand what happens here. For that, in order to do that, we're going to have to enlarge this area, one area, and we're going to enlarge only one myofibril. And one single myofibril will have this particular feature. Let's go like that. I'm going to come this way. Now, as you probably know, these myofibrils are composed of filaments of actin and myosin, which give these characteristics. I'll try to come here this way. It's probably better. The thick filaments are the thick filaments of myosin. And they are drawn different color. The thin filaments, I'll draw a different color. Perhaps yellow will give you the impression of being lighter. The thin filaments are going to be the filaments of actin. In other words, the area where the thick filaments of myosin are is darker than the area composed of the filaments of actin. In other words, like this. It goes like this, and so now these fibers that are numerous in each myofibril, they are joined together here, right? And also here. Now you have to memorize some letters for each band, and that's unfortunate, but that is exactly what we have to do. Okay, this dark band is going to be the A band for anisotropic, and this one will be called I for isotropic, the lighter corresponding to the filaments of actin. Now, we do have other numbers, letters to remember. For example, the band that joins together the filaments of actin is called the Z line. And the one that joins together the myosin filaments is called the M band. Now, what happens why do we have this peculiar, so orderly, incredibly ordered arrangement? Because these filaments have to slide between each other in every muscle contraction. So this is the reason. That way, for example, the yellow filaments or the actin filaments will go in, and there's an extra little area called the H, the H band for hell in German, which is clear. That way you are getting an idea of why the skeletal muscle is like that. Excuse me. Now I'm going to try to share with you guys why this structure is set like this in order to receive the deposits of calcium, if you want. The calcium will be, I'll use a different color. The membrane, the membrane has tubules that go below and surround each of these structures. Let me put it like this. Each one of these filaments and liberates calcium. So when the impulse, I forgot to tell you that the impulse, this skeletal muscle doesn't work by itself. There is a neuron, a motor neuron, a motor plate that was going to give the order to the muscle to contract. And once there is depolarization of the membrane, calcium ions will be released. Now, the famous T-tubules are structures that are simply tubular invaginations of the membrane that surround in different places, mostly at this moment at the junction of the I and the A bands. In addition to that, we'll have the sarcoplasmic reticulum. We have another cisterna, okay, which is the terminal cisterna that communicates to the other cisterna. In other words, if you try to simplify this, we're dealing with an invagination of the membrane forming a circle around every single myofibril at the junction of the A and the I bands, and the calcium will be released, and the contraction of the muscle will take place because of the fact that the filaments get together. Now you can ask yourself, well, how really happens? We have an idea, we have a molecular idea. Molecular biologists would probably have a better idea than I do. But the filament of myosin, let me see, is formed like this, like mini golf kind of the filament of myosin is formed by a longitudinally arranged portion and another like this in several different molecules. These units, these rounded units of the myosin, are going to try to get in touch with the actin, and once they get in touch, they will move. They will move like this. Now, what about the actin? We said that the actin is here, and it is like little beads. I'm going to try to be simple. It almost forms like a beads, like two chains, has beads that are meant to be in contact with the myosin. The only problem, well, it's not a problem, but we have another complex called the troponin complex and tropomyosin. In other words, there is another molecule attached, and surrounding the actin is the tropomyosin. And there is another complex called troponin that binds calcium. It binds calcium. In other words, every time the calcium attaches here, the molecule of actin will move, and the tropomyosin will liberate the place where the troponin, where the myosin can grab. So in other words, the muscle will take place when the calcium is there and attached to the troponin complex and the tropomyosin, liberating the sites in the actin filament. It's very simple, right? It's obvious. Now, before going to the smooth muscle, I'll just share with you guys that scientists have discovered a way to find in the meat, in the muscle, the skeletal muscle of humans and also in animals, beef, etc., the stem cells. Apparently, they are very simple. They almost look like fibroblasts, and if they use it, they put it in a culture medium. Those cells will grow and grow and grow and produce what? Produce meat. In other words, people are producing meat from stem cells coming from an animal. And according to what they say, they don't need to kill the animal. They just do a little anesthesia, I suppose, and get a biopsy of muscle, and they harvest those stem cells, and then now they put it in a medium, and then voila, you have a chicken meat or beef meat that has been made in a laboratory. That's something I wanted to share. Now I'm going to go quickly to share with you guys something on the smooth muscle. Well, when it comes to skeletal muscle, everybody remembers where their muscles are, right? The smooth muscle is more internal and mostly associated with viscera. For example, if you come to look to the uterus will have this like a pear-shaped and has in the center a cavity, right? Which has this endometrium. And where is the muscle? The muscle is, cervix doesn't have too much muscle. It is the rest of this is the smooth muscle. And also there is many other, what is the outer portion? The outer portion, let's put it a different color, would be the peritoneum. And then you have the uterus made essentially of smooth muscle. Then you have other organs, I would say, like arteries, or even the digestive tract. They have a mucosa or have an endothelium. And here's where we have the smooth muscle. In the vas deferens, in the ureter, the bladder, so many internal organs. And The smooth muscle is smooth. In other words, gives the impression of absence of striations. And you know pretty much how it looks. It's almost like this. And the whole cytoplasm is homogeneous, although it contracts, and although it has filaments of actin and myosin. So in other words, I'll try to make it, the nucleus is kind of almost cigar-shaped. And this cell will usually obviously it will not take the entire, it's small, and it goes along with other cells, almost like a school of fish, like this, pretty much, kind of a little bit, almost like this, trying to go the more thicker area with the other area. Now, every time the muscle contracts, it will become a little bit rounder, right? So we'll have, H1 has its own nucleus. I will not paint the cells because you can imagine that. The cells, they don't need the autonomic nerves to tell them when to contract. They contract by themselves by local influences or hormones, but they are also influenced by the parasympathetic. Why is the smooth muscle smooth? Simply, let's try to use this other one. Simply because the filaments of actin and myosin are pretty much crisscrossed like this. They are not forming fibers, but they are forming like a crisscrossed pattern. They go from an adhesion density here to a focal density here, adhesion density, focal density, etc. And that is pretty much the way it is. Tumors of the smooth muscle tumors are very common. I'm just going to mention for sharing with you guys. Usually in women may have a leiomyoma, a nodule there, which is called fibroid, and it's very, very common. Now we'll go and try to make it easy and shorter. We're going to go to the heart muscle. The heart muscle, we'll have to think about that the heart has a heart shape, if you don't tell you, with the big vessels here, and the cells are going to be concentric around this hollow muscle, if you want. The heart is a hollow muscle, and the cells are elongated like this. I'm going to change the marker because this one is probably better. Like cylindrical, except for the fact that at the end they branch. They branch. In other words, they are cylindrical but branched. And how do they get? Do they contact the other cells? Is at the very end. So we have here structure called intercalated discs that make it possible for each cell to be connected to the other cell. The cell is not multinucleated, usually has a single nucleus, but it maybe have two. And it's kind of in a little bit almost rectangular in shape, it's pretty dark. And obviously, we are going to have striations. Why do we have striations in the muscle heart? Because there is numerous myofibrils, just as the skeletal muscle. But these fibers are not so, regularly arranged. But anyway, when you look at the skeletal muscle, the cardiac muscle, excuse me, you see that the heart muscle is striated the same way. Now, we called these structures intercalated discs, are essentially dark areas, a little bit darker areas, that are points of union between two cells, like this, like finger with the finger, and are composed of pretty much fascia adherents. It's a little bit almost like a sonnular adherence, but longer. Also, we have desmosomes, and also gap junctions that allow the conduction to proceed in a fast fashion. Now, as you know, probably know that the heart has specialized muscle cells that produce its own impulse. In other words, that impulse, which is in the right atrium, will extend to the entire heart, and the heart will respond in a very simultaneous fashion, thanks also to the fact that we have specialized conductive cells called Purkinje cells. I think with this we can actually end. I apologize if this is a little bit kind of a messy, but this is a kind of a little bit complex subject. I hope that you guys get something out of it. See you next time.